This is another series of our biofusion seminar series, and then today I believe it's more like another technologies and latest stuff. So I give my mic to our organizer so he can actually preside and he can actually organize this presentation and panel discussions. Okay, it's all yours. organizer 시작하시면 됩니다.
교수님, 제가 준비를 했어가지고 제가 시작하겠습니다.
그래요.
네.
오케이, 그렇게 하세요. 네.
네네. Nice to meet you, everyone. I'm moderator Jinyan An, and I'm in Professor Jeongkyun Choi's lab. My research area is about developing prediction models for immunotherapy responsibility using NGS genomics data. Today, our group will introduce about what is NGS technique and its application to the microbiome field. I think many of you guys have heard about NGS technique, but some are not familiar with this technique. So Jung-Eun Lee will briefly introduce about basic concept and mechanism of NGS, and Jinong Yang will present you about how this technique could be applied to research field. After presentation, our panelists Kyung-Han Han, Jason Park, and Kyung-Hee Kim will discuss about how NGS could be applied to disease treatment and commercialization of NGS and limitation of NGS technique. Please enjoy our seminar, and if you have questions, please write on our Q&A board. First, Jung-Eun Lee will present about mechanism of NGS technique. Jung-Eun, please start your session.
Hi, my topic for today's seminar is next generation sequencing. I will introduce this topic in broad terms. First, what NGS are, where they are being used. Second, how sequencing technology has developed over time. Lastly, I will compare these technologies and introduce how they will develop in the future. Next generation sequencing analysis is a fast analysis method of sequencing genomes, also called high throughput sequencing or second generation sequencing. DNA sequencing is a technique that analyzes the sequence of bases that make up the DNA chain over cells of life in a biochemical way. Sequencing technology evolved over the past 15 years, and new methods are continually being commercialized. As the technology develops, so the increases in the number of corresponding applications for basic and applied science. As shown on the screen, NGS can be used to analyze DNA and RNA and even protein. It is used in various places, including personalized medicine implementation through genomic research, lipid and accurate diagnostic technique development, DNA profiling in forensic science, and analysis for uniform cell culture in industry. This is timeline of sequencing technologies. Sequencing technology, which develops so fast and is frequently used in many places, was first started by Sanger. And since 2005, sequencing technology has developed rapidly, reaching second and third generation technology. Now, let's find out more about NGS technologies. The founding methods in DNA sequencing were the Sanger deoxy synthesis and Maxim-Gilbert chemical cleavage. Since Sanger sequencing is introduced in a more standard way than Maxim-Gilbert chemical cleavage, so I will focus on the Sanger sequencing here as well. Sanger sequencing uses specific chain terminating nucleotides, the deoxynucleotides that lack a 3' OH group. Thus, no phosphodiester bond can be formed by DNA polymerase, resulting in termination of the growing DNA chain at the position. The DNT pits are radioactively or fluorescently labeled for detection in sequencing gels or automated sequencing machines. The Sanger sequencing method was developed in 1977. Although relatively slow by current NGS standards, improvements in the Sanger chain terminator methodology, automation, and commercialization have enabled it to remain the most appropriate sequencing method for many current applications. Specifically, the development of automated, refillable, reusable capillaries have contributed to the enhanced speed and ease of the Sanger process. Also, Sanger process can give high-quality sequence for relatively long fragments of DNA. On the other hand, it is an expensive process with a low data output. The need for simpler and faster sequencing processes lead to the development of new technologies for DNA reading, collectively named next-generation sequencing. The term next generation has implied a next step in the development of DNA sequencing technology and suggests there will be a next generation naming of new technologies in the future. So, next generation sequencing is called second generation sequencing. Sequencing by hybridization so next generation sequencing is called second generation sequencing. Second generation sequencing methods can be grouped into two major categories. Sequencing by hybridization uses a array of DNA oligonucleotides of known sequence on filters that were hybridized to labeled fragments of the DNA to be sequenced, and then wash away the unwanted non-hybridized DNA. It depends upon using specific probes to interrogate sequences, so mainly used in diagnostic applications or identifying disease-related SNPs. Sequencing by synthesis are a further development of Sanger sequencing without the deoxy terminators. In combination with repeated cycles of synthesis, imaging, and methods to incorporate additional nucleotides in the growing chain. There are several platforms according to the technical details of the sequencing reaction. These platforms have the same process: sample preparation, cluster growth, sequencing, and data analysis processes in common. But there are slight differences in sequencing methods. Pair sequencing detects pyrophosphate, a byproduct of nucleotide incorporation. The first process is sample preparation and cluster generation. Individual DNA fragments 400 to 700 base pair long are aggregated to adapters and amplified by PCR in an individual immersion bead reaction. DNA sequences on the beads are complementary to sequences on the adapters, allowing the DNA fragments to bind directly to the beads, ideally one fragment to each bead. The next process is sequencing and data output. DNA synthesis followed by chemical detection of the DNA synthesis directions then occurs in a picoliter-sized chamber where pyrophosphate release is measured. By consecutively flooding the chambers with sequencing reagent containing one of the four nucleotides, when the correct nucleotide is incorporated in the synthesized strand, pyrophosphate release is measured utilizing a light-generating reaction. Ion semiconductor sequencing is a method of DNA sequencing based on the detection of hydrogen ions that are released during the polymerization of DNA. The ion-turned method, like the pure sequencing method, attaches an adapter to the DNA fragment and amplifies it to create a cluster. And in the next process, this method directly converts nucleotide sequence into digital information on a semiconductor chip. In a DNA synthesis reaction, when a correct nucleotide is incorporated across from its complementary base in a green DNA chain, a hydrogen ion is released. This changes the pH of the solution, which can be recorded as a voltage change by an ion sensor, much like a pH meter. Illumina sequencing is based on a technique known as bridge amplification, wherein DNA molecules with appropriate adapters located on each end. It contains oligonucleotide sequences complementary to a ligated adapter. Sequencing by reversible terminator chemistry uses reversible terminator bond the antipiece in a cycling method that comprises nucleotide incorporation, fluorescence imaging, and cleavage. A fluorescently labeled terminator is imaged as the antiP is added, and then cleaved to allow incorporation of the next base. These nucleotides are chemistry blocked such that its incorporation is a unique event. An imaging step follows its base incorporation step, then the blocked group is chemistry removed to prepare each strand for the next incorporation by DNA polymerase. In contrast to second-generation sequencing methods, third-generation sequencing methods aim to sequence long DNA molecules. It is possible to pass long DNA molecules through small diameter holes and measure differing currents as its nucleotide passes by a linked detector. Two types of nanopore systems for DNA sequencing are being developed: biological membrane systems and solid-state sensor technology. Biological nanopore sequencing relies on the use of transmembrane proteins embedded in a lipid membrane to produce the pores. A single protein nanopore is integrated in a lipid bilayer across the top of a microbial equipped with electrodes. Solid-state sensor technology uses various metal or metal alloy with nanometer-sized pore that allow DNA or RNA to pass through. This method is considered to be next-generation nanopore technology because they bypass the use of organic supports and are more stable. A summary of the sequencing techniques we have seen so far is as follows. First and second generation technologies are sequences using many DNA copies, and the length of the readable sequence is not long. Third generation technology, on the other hand, can be sequenced with a single molecule and has a long readable sequence. It can also be seen that the cost of sequencing technology is decreasing in general with third generation. As DNA sequencing technology advances, the goal will be faster and more accurate sequencing and lower error rates and minimal artifacts and input DNA and RNA at lower cost. At the front end, advances in robotic and liquid handling sample processing will contribute to this advancement. Equally important will be advances in faster and more accurate bioinformatics data analysis, as well as data transfer and storage. Thank you.
Thank you, Jung-An. It might be helpful for understanding history and basic concept of NGS. Through a comprehensive and good explanation, it was easy to interpret the concept and mechanism of NGS technique from Sanger to third-generation sequencing. And next, Jinok Yang will present about how NGS technique could be applied to the microbiome field. Jinok, please start your session.
Sure.
화면
요거.
네. 그래서 PDF가 어떤 거죠?
데스크탑에 있는데.
한번 해보실래요?
근데 왜 연결이 안 되는 걸로 돼 있지? 됐나요?
안 돼 있어.
어떻게 해야 되지?
Sorry, there was some problem to share the PPT. 그러면 지금 뭐 그냥 메일로 저한테 보내주시면 제가 쉐어를 할까요?
그게 좋을 것 같아요.
네, 그게 빠를 것 같아가지고.
잠깐만요.
저 전달해 주시면. 일부러
맞죠?
네, 한지 맞습니다.
I am second presenter Jinong Yang. My talk is about microbiome and applications in biomedical area. Today, I'm going to introduce some application cases in the representative human microbiome project. In this figure, we display the timeline of sequence-based metagenomics studies and shows the range of environment between 2003 and 2017. This is an attempt to also highlight the advancement of sequencing techniques over time and provide information regarding appropriateness for applications in exploring the human microbiome and metagenomes of other diverse environments. To know what kind of a microbiome and what they do, these various platform techniques are required. Complex microbiome studies cover various areas, starting from the level of complete microbial cells, followed by DNA, RNA, protein, and metabolites. In that order, the focus of the studies shift from microbial potential over the metabolic potential
towards microbial functioning. To find out the microbial potential function, Human Microbiome project has been started. The Human Microbiome project is as a logical, conceptual, and experimental extension of a human genome project. Human Microbiome project is to improve understanding of a microbial flora involved in human health and disease. This is the overview of phase 1 and 2 of a Human Microbiome project. Phase 1 focused on characterization of a community in different body sites, skin, oral, and gut. Phase 2 focused in the integration of multi-omic data from host and microbiome, drawing correlation with diabetes and inflammatory bowel disease. Through Human Microbiome project, we find how many kinds of bacteria is all over the body. The 10 to 100 trillion number of symbiotic microbial cells harbored by each person, primarily bacteria in the gut that make up the human microbiota. 80 to 90% individual humans are different from one another in terms of the microbiome. Thus, to conquer human disease and cancer, we need to know the human microbiome. I will say I will provide some studies based on microbiome. The case 1, pregnancy and preterm birth. This project is concerned with the role of microbiome in the occurrence of preterm birth, which account for nearly 10% of all births and constitute the second leading cause of neonatal death. Case 2, onset of inflammatory bowel disease, IBD. IBD studies focused on understanding how the gut microbiome changes in adults and children from IBD. IBD is inflammatory autoimmune disorder that manifests as either Crohn's disease or ulcerative colitis. Through this study, they characterized the gut microbial ecosystem for diagnosis and therapy in inflammatory bowel disease. Case 3, onset of type 2 diabetes. This is to perform a longitudinal analysis on the biological process that occurred in the microbiome of a patient and risk for type 2 diabetes. They identified the molecules and signaling pathways that play a role in the etiology of disease. Case 1 to 3, these 3 examples of microbiome associated human conditions are available to explore the behavior of a human microbiome over the course of complex conditions. In additionally, case 4, the gut microbiome and schizophrenia study. Diabiosis could exacerbate inflammation in the disorder through increased intestinal permeability. Indeed, elevated level of bacterial translocation marker, CD14, have been observed in schizophrenia. The existing evidence for this biosis as a factor in the pathogenesis of schizophrenia is underwhelming. The use of probiotics as a treatment in schizophrenia aimed at improving side effects of antipsychotics and complementing their action, especially in terms of cognitive impairment. Case 5, the interconnections among dose and microbiome-associated biomarkers. In this example, several environmental exposures and conditions can alter microbiome diversity and metabolism. These microbiomes participate in biotransformation and interact with image, therefore transduce the responses both to and from the host organism. The integration of a microbiome into the environmental health paradigms will broaden and amplify our concepts of susceptibility. The microbiome-associated biomarkers are known to play important role in the studies of metabolism and its outcomes, such as autoimmune disease, asthma, and metabolic syndrome. In the light of these downstream connections, microbiome-associated biomarkers can serve several purposes. Recently, from several microbiome studies, research results confirm the therapeutic effect through the improved strain, the engineered microbe. Disturbance in the gut microbiota has been described in several cancers through inflammatory and immunological interactions. This table shows the the time-linked global microbiome-based drug development company. The microbiome biomarket is growing rapidly. It is expected in the diagnostic, treatment, development sector in the future. Consequently, microbiome signature as a biomarker valuable future medical paradigm of personalized treatment and proactive prevention of management. So listen to the microbiome. The microbiome is very important, especially microbiome signature as biomarkers. They play a role in human health and disease. And new drug development, specific disease, autoimmune disease, gastrointestinal tract, diabetes, obesity, cancer, etc. The mechanism of action of microbiome. This is a good resource. So microbiome signature very valuable and important. Okay, so microbiome is a new approach in medical research. Thank you.
Thank you, Gino. Through your presentation, we could see how could this field-to-field could be synergistically cooperated, and what is the human microbiome project, and how it could be used to find the biomarker study. Before we go to next session, thank you for your waiting before we present our presentation because caused by the computer error. So then the first presentation session is ended, and let's move on to the discussion session. In discussion session, first, Kyung-Han Hwan will discuss about how NGS technique could be utilized to the field of disease treatment. Kyung-Han, please start your session.
Hi, I'm Kyung-Han Han. I'm gonna present about exploring the human microbiome using NGS for effective diagnosis and therapies. This is a table of contents of presentation. Here are 3 parts. Why is the human microbiota important? The microbiome as therapeutic target, and 2 examples about real application cases. First, why is the human microbiota important for diagnosis and therapies? Because humans are not alone. From the moment we are born, millions of microorganisms populate our bodies and coexist with us. The interaction between the human microbiome and immune system play a key role in determining the wellness or disease status of the human body. So these biases may lead to a plethora of diseases, including skin, inflammatory, metabolic, and neurological disorders. A better understanding of the host-microbe interaction is essential for determining the diagnosis and appropriate treatment of this ailment. Second, the microbiome as therapeutic target. Using NGS technologies, comparative analysis between metagenomes can provide additional insight into the function of complex microbial communities and their role in host health. The significance of the microbiome on host health has led to the emergence of new therapeutic approaches focused on the prescribed manipulation of the host microbiome, either by removing harmful taxa or reinstating missing beneficial taxa and the functional roles they perform. Then how can we applicate these approaches? There are some methods. For example, dietary interventions, multi-species microbial supplements, fecal transplantations, and drug treatment. These approaches to modify gut microbiota generally focus on depleting overabundant members or overall microbial load. I introduced two examples that show how these approaches can be used. The first example is CDI, Clostridium difficile infection. Clostridium difficile infection is associated with broad-spectrum antibiotic therapy, and it is the most common cause of infectious diarrhea in hospital patients. That means Clostridium difficile infection involves an initial disruption of the normal colonic bacterial flora by antibiotics. Treatment of recurrent Clostridium difficile infection with antibiotics lead to recurrence in up to 50% of patients. Rather than antibiotics, restoration of the normal colonic flora may allow elimination of Clostridium difficile and clinical recovery. This is a result of fecal transplantation experiment. In this paper, researchers used fecal filtrate transfer method to prevent uncontrollable risks for infectious and metabolic diseases due to transferring undefined living bacteria, particularly in immunocompromised patients. They collected fecal samples from patients before and at 1 week and 6 weeks after FFT. Microbiome profiles of donors and patients are compared using NGS. They confirmed that FFT restored normal stool habits and eliminated symptoms. In this figure, distance between symbols on the plot did not relate these similarities in bacterial community. You can see patients' bacterial communities are mapped near donors one after FFT. The second example is rheumatoid arthritis. Human microbiome is not only involved in the digestion and absorption of food, but they can also exert a protective function by preventing adherence of pathogenic bacteria to the mucosal layer, and they play a pivotal role in modulating the innate and acquired immunity of the host. Recent advances in sequencing technologies enable the investigation of these biases, which could be involved in the onset and maintenance of different chronic autoimmune diseases. In RA patients, an accumulating body of studies demonstrate a pathogenic role of dysbiosis of the oral microbiota. Alterations in the gut, dental, or saliva microbiome distinguished individuals with RA from health controls are correlated with clinical measures and could be used to stratify individuals on the basis of their response to therapy. In this paper, researchers used drugs to identify compositional and functional alterations in RA-associated gut and oral microbiomes that are partly relieved by disease-modifying anti-rheumatic drug treatment. And next, paired and metagenomic sequencing was done on the Illumina platform. As a result, the researchers found that hemophilus are depleted in individuals with RA at all 3 sites and negatively correlate with levels of serum autoantibodies, whereas lactobacillus and salivarius was overrepresented in individuals with RA at all 3 sites and was present in increased amounts in cases of very active RA. These findings are a first step toward microbiome-based therapeutics and patient stratification in preclinical and clinical phases of RA. This is the references for this presentation. Thank you.
Thank you, Kyung-han. It was interesting in aspects of practical application of this technique, and we could see details and values of microbiome combined with NGS analysis. Also, Jason will focus on commercialization of NGS technique, which is very big part for its spread on academic and industry application. Jason, please start your discussion.
Okay, so our previous panelist has discussed about the successful cases in medical research. And so I would like to focus on business field of microbiome therapeutics. Currently, the microbiome market is mainly focused on wellness products, including probiotics and prebiotics, is occupying 83%, and only a small portion is dealing with therapeutics and diagnostics. However, if you look at the table by BCC Research, if we follow the increasing trend of the market size increase, then the market size will increase by $9,000 million within 5 years. So the market size is quite good. So and also, if you see the statistics for microbiome therapeutics, the top 10 clinical fields that the microbiome covers is gastrointestinal diseases and infectious diseases, which is quite intuitive and reasonable. But there are some striking fields that microbiome also covers, which is oncology and even central nervous system. And there's so many different types of medical fields that microbiome can cover. And if you see the next chart, which is classified into a developmental stages of the therapeutic pipelines, most of the pipelines are still in preclinical stages, but about 50 cases are in phase 1 till phase 3. So probably within 5 to 10 years, microbiome therapeutic products are not anymore a fairy tale. We can also classify the microbiome therapeutic into 3 different types of fields. The first group is having quite solid proof of concept results. So many different companies are actively developing the drugs, applying microbiome concepts. And it includes gastrointestinal disorders, infectious diseases, metabolic disorders, skin disorders. And one good news is that they are dealing with some rare diseases, including hyperoxaluria, urea cycle disorder, phenylketonuria. And I think this is quite good news that many big pharma is quite hesitating of dealing those rare diseases because of the profit, but those main microbiome products are focusing on those rare diseases, which is good news. The second group is having decent amount of proof of concept results, but not solid. So the products are emerging nowadays, and it includes obesity, which is healthcare problems, and cancers. And cancer is not only including the combination therapies with chemotherapy, but also with immunotherapy. And the third group is having minimal proof of concept results, and they're very in a preliminary stages. And it includes neurological disorders, cardiovascular diseases, and inflammation, including all kinds of autoimmune diseases like arthritis. So anyway, if you look at this chart, there are so many different fields that are covered by microbiome therapeutic fields. And this diagram is showing the representative therapeutic companies, and we can classify those therapeutic fields into 2 parts. First one is microbiome composition control, and the second one is microbiome human interaction control. In order to control microbiome composition, the microbiome therapeutics company is actually applying the prebiotics. It's actually giving a food to bacteria in our guts. But we can actually add the microbiome itself, which is called probiotics. And it can be either introduced by drugs, but also directly transplanting to fecal matters from the normal persons to the patients, so that the probiotics can deal those symptoms we want to target. And also one of the big issue is that the probiotics, if we find a good bacteria, then we need to be able to culture the single strain so that we can introduce only that bacteria but not others. So the company called Evelo and Synlogic is trying to do that. And also you can subtract the microbiome, the harmful flora of bacteria. And a company called BioMX is actually trying that. If we look at the second sector, dealing with microbiome human interaction control, the company called Pylum Biosciences is actually applying the bacterial metabolites, and it's I heard it's dealing with the issues of gut-brain axis, as the previous presenter has been explained. And not only these representative companies, there's so many different companies, dealing so many different kinds of medical fields. And I think this is the interesting part, the sensational part of microbiome. It's not only dealing with like gastrointestinal disease, which is what we previously thought that microbiome could be applied, but It can also deal with all different kinds of unpredicted medical fields. Of course, microbiome therapeutic is also a drug development process. So it requires a tremendous amount of money. So those startup companies are actually backed up by very big global big pharma or global big food industries like Johnson and Johnson, Pfizer, and Nestle, and yeah, they're having a very good synergies in terms of drug development process. In Korea, not many companies are developing microbiome therapeutics, but there are 3 quite successful cases, including Co-Biolabs. They're dealing with autoimmune diseases. They're quite globally, they're quite, global companies, And Genome and Company is trying to deal with the cancer therapeutics with microbiome. And Chunlab is currently dealing with healthcare products. They're profiling daily microbiome profiles of in the fecal matters of human. And actually they are dealing, they want to tackle those healthcare issues with prebiotics and probiotics, personalized prebiotics. So unlike the traditional omics technology, like genomics and transcriptomics, which was very successful in research area, but was having a huge problem when it has to be translated into a medical field. But I think microbiome is, if it has some kind of good findings in the research field, then it can also be translated into medical field. It can also be the solution as well. So that's why I think the microbiome therapeutics can be the game changer of bioindustry in a relatively fast paces. So just let's look at what happens, and I hope you are ready for the new trend. Thank you.
Thank you, Jason. Through your discussion, I think we could identify the present stage of microbiome and surprised because there are so many companies to try microbiome in therapeutic purpose. I think in future, we could imagine that these clinical trials will be actively utilized in practical areas. Okay, it's our last discussion, and Kyung-hee Kim will discuss about limitation of NGS technique, which we could keep in mind. Kyung-hee, please start your discussion.
Hi, I'm the rest panelist Kyung-hee Kim. The previous panelists gave good presentations, but I have the opposite thought with the previous panelists. I think there can be some practical problems currently to use NGS in clinical application. Although there is a desire to use NGS in clinical applications such as microbiome therapeutics, significant limitations currently exist. These limitations include sample quality, library preparation, difficulty to sequence some areas, and Illumina's dominance and commercialize. First, sample quality. In NGS, samples of low or variable quality can corrupt downstream processes such as library preparation and ultimately confound analysis. Real-world samples often present much more of a challenge because extensive DNA damage can be caused. There can be tremendous variability in the amount and types of damage in sample DNA, such as inter and intra-strand crosslinks, breaks, accumulation of single-stranded DNA, and other forms of DNA damages. And the variable amount and types of damage can negatively affect the final result. Next, there are some limitations in library preparation. Although the major sequencing platform companies have spent years bringing down the cost of generating raw sequence, the same has not been true for library preparation. Library preparation for human whole genome sequencing at about $50 per sample is still a relatively minor part of the total cost. But for other applications such as sequencing bacteria genomes or low-depth RNA sequencing, it can account for the majority of the cost. Several groups are working to bring the effective cost down, but there haven't been many developments on the commercial front. And there are some difficulties to sequence some areas. Homologous regions, repetitive regions, and gist-rich regions are not reliably interpretable by the current NGS platforms. Homologous regions, including pseudogenes, are areas of the genome with high sequence similarity. Fragments of DNA that are sequenced from the target gene and homologous regions may be so similar in sequence as to be indistinguishable. And the shorter the length of sequence, the more likely this is to occur. So, fragments of DNA from a target gene and homologous regions will have poor mapping quality, and reads belonging to the homolog can be mismapped to the real gene. For example, in this figure, the right side shows the target gene and the left side shows the pseudogene. As you can see, a similar number of sequencing reads are being mapped to the pseudogene as to the real gene. Therefore, these reads would have a low mapping quality score. Also, NGS market is almost dominant to Illumina now. Illumina's dominance has meant that the vast majority of the data is based on short reads. There are some advantages and disadvantages of short reads and long reads. Having a large number of short reads is a good fit for a number of applications, such as detecting single nucleotide polymorphisms in genomic DNA because of its high accuracy and throughput. However, short reads can only access a fraction of genomic content because genomes are substantially repetitive and much of the information in the genome is encoded at long scales. So, short reads alone are insufficient in a number of applications, such as reading through highly repetitive regions of the genome and determining long-range structures. However, to get the most out of these long-read platforms, it is necessary to use new methods for the preparation of DNA samples. Lastly, it may be difficult to commercialize. With NGS, the sheer amount of data will be generated. For example, the vampire for a single human whole genome sample is about 90 gigabytes. So, a relatively project of 100 samples would generate 9 terabytes of data per year. So and with a single Illumina high sequence instrument, is capable of generating of 130 terabytes of data per year. So the storage can quickly become a concern to commercialize. Also, it requires fast data processing, and although many institutions may have ability to purchase NGS equipment, many lack the computational resources and staffing to analyze and clinically interpret the data. Like this, there are many limitations to using NGS in clinical applications now. There can be low sample quality by DNA damage or sequencing error. For many of the identified abnormalities, the clinical significance is currently unknown. Next generation sequencing also requires sophisticated bioinformatics systems and fast data processing and large data storage capabilities. And it can be costly. And many institutions lack the computational resources and staffing. Therefore, I think significant limitations currently exist to use NGS in clinical applications. Thank you.
Thank you, Kyung-hee. I think through your discussion, we could keep in mind that to successfully apply NGS technique in microbiome and many other areas, there are so many considerations from sample quality to other many details. Although we couldn't predict the future, through considering these many limitation factors, we could overcome these things successfully. Thank you again for your nice presentation and discussion. And after this, we will go to the Q&A session. While listening to our seminar, if you have questions, please write on our Q&A board. Then I will read and our group will answer it.
While students are preparing, I have one question for one of the speakers, I believe. So some of the speaker mentions about the commercialization of microbiome, right? So I'm just wondering that for example, FDA approval process for microbiome product, is it as strict as like other small molecule drugs or other like protein drugs, and how does it go? Can you explain that?
I think I can answer the question. As far as I know, the approval process is exactly same as all different kinds of chemical drugs. But one thing to consider is about patent issues, because in theory, the organism itself cannot be patented, but the culturing method or the metabolites purification from certain strains can be patented, those technologies. So I think the major issue in this industry is about patenting specific technology that can be commercialized. And yeah, so.
Okay.
Is there any other questions?
Maybe I have another question then for you. So I believe one of the especially the microbiome and latest stuff, one of the target disease is quite related to the autoimmune disease. So I'm just wondering why this kinds of microbiome or those kinds of things is effective in dealing with the autoimmune disease. Is there any kinds of like a specific biological reason?
Oh, should I answer again? Okay. As far as I know, I think so. Autoimmune disease is mainly regarding to inflammation, and inflammation can be caused by many different kinds of factors, but they're dealing with the harmful chemokines or metabolites that are secreted from the bacteria that might cause those abnormal activation of immune systems. As previous presenter explains, if the gut permeability has been increased, then those chemokines or metabolites are leaking out of the guts, and it flows into the blood and may affect those nervous system. And altogether, that can stimulate immune system, leading to some kind of abnormal behavior. Yeah, that's what I know.
Okay, great, thank you.
I think there are no other student questions. So is it okay to end our seminar? Okay. Our seminar is ended, and thank you for your attention.
Great, thank you very much.